Hello. So for those of you who might have missed my introduction before, my name's Meg. I'm a second-year PhD student at Kingston University, London, and I'm also chairing this conference. I'm going to try to keep this presentation brief because you guys will be hearing a lot of my voice today. So my research focuses on efficient language modeling for non-coding variant effect prediction from human DNA sequences. I realize that's a bit of a mouthful, so let's break it down. If you can see my slides, that is. Great. So we all know that genetic variants impact disease risk and treatment response, but existing research mainly focuses on variants within genes, so coding variants. But there's actually a lot of importance in non-coding variants too. The non-coding region of our genome plays a huge role in gene regulation, and so any variants here can have a massive impact on disease risk and treatment response too. But non-coding variants are underexplored and they're a lot harder to interpret because they affect genes through different mechanisms such as regulation, or for example, a variant might be much further away from the gene that it impacts. For instance, you might have variants somewhere over here impacting a gene somewhere over here. So how can we interpret these things? Well, this is where LLMs or NLP come in. So transformer-based large language models are the basis of things like ChatGPT, but they're also really valuable in genomics research. They're great at kind of uncovering these context-dependent relationships within DNA and between variants and genes. But the issue with transformer-based LLMs centers on the fact that the attention mechanism, which is central to those LLMs, scales quadratically with sequence length. So what this means is that as you increase the sequence length of, for example, DNA sequences that you put in, the amount of time and computational resources required for training and testing these language models increases quadratically. So this is a huge problem for a lot of kind of tasks that we try to solve, but particularly for non-coding variant effect prediction, because we have these long sequences and we're trying to predict the links between, variants here and genes there. So how can we solve this? So one of the ways we can look at tackling this issue is with pruning and knowledge distillation. So if any of you guys are in the machine learning field, you might have heard of pruning before. So this basically just involves cutting up parts of our neural network, or like in this case, our LLMs. So you can do something called depth-based pruning, where you remove whole layers. You can do width-based pruning, where you remove neurons, or you can move on to things like distillation, where you minimize certain losses during the training process to reduce the size of your models and hence make them more efficient. So I'm showing a preliminary study here where we used a large language model called DNA-BERT2. So this is pre-trained on multi-species genomes and consists of 12 transformer layers. So this has performed really well on some other tasks, for instance, COVID variant classification. So we decided to try it out for our problem. So one of the key things to understand with LLMs is they learn a lot of different things, but not all layers are created equal. I think my table is cutting off a little bit here, but we found that removing different layers of the LLM really changed the results in different ways. So removing some layers had a larger effect on our results, whereas removing some layers had a negligible effect. And you can see that after we've removed the first 6 transformer layers, actually our accuracy didn't go down that much. So this suggests that perhaps like those layers aren't as important in the equation. So because this worked quite well with a problem that they showed in the paper, we decided to try it on our non-coding variant effects prediction task. So what we were trying to do here is a binary classification of these variants as pathogenic or benign. So again, we looked at removing individual layers, and you can see that for different layers, again, like the accuracy in the AROC on our task really varied. So this graph is showing like which layer was removed, and then the accuracy in AROC for these. And you can see our baselines are here. And you can see that with removing these last few layers, the accuracy was actually pretty similar to what we had originally. And again, after removing the first 6 transformer layers, we've actually still got a pretty decent accuracy, and actually better than we had with removing individual layers. So these preliminary results show that pruning can actually be quite an effective way to keep the results while really reducing the computational cost. So you can see that this reduced pruning half of the transformer layers reduced our number of parameters down to about half. It reduced our inference time, and crucially, it also really decreased our training time to about half. So you can see that's a lot more efficient. It pretty much preserves the results. So this could be a pretty good way forward. So our conclusions here are that not all LLM layers are created equal. Tactical pruning of layers can decrease computational costs while maintaining performance. And also removing contiguous layers, so our first 6 transformer layers, can actually produce better results than removing individual layers. And this reflects existing research. So my next steps with this research are going to be to figure out what exactly is being learned by these different LLM layers and work a bit on interpretability. I've also got a poster on this at the VeriAI track tomorrow, so feel free to come chat to me there as well if you'd like to. And if you'd like to learn a bit more about LLMs for variant effect prediction, I've got a review paper about that coming out very soon. But thank you guys very much. I'm sure you all sick of hearing my voice already, so I'll wrap this up. Thank you.
All right. So we're actually going to do the break right now, the coffee break. It's going to be a 15-minute coffee break. And then we're going back to Guillermo Castello, that is the next speaker. So we're going to do that after the coffee break, if everyone agrees. So we'll see you here again in 15 minutes.
Sorry, guys, I'm back. I was just going to say, feel free to pick up your free t-shirts at the registration desk there. And yeah, I'll see you after the coffee break. Thank you very much.
And please tag us in your pictures in social media.
Yes, we have a hashtag SCS 2025. Thank you.